Dr.
Can you hear me?
He's, by the way, he's Wolverine of the Biome Squad. See the face
Okay,
and
yeah.
everything?
He definitely looks like Wolverine. That's
He's Wolverine. I mean, like his position, everything.
yeah,
In,
so who am I?
government.
I'm Richard Dreyfuss, the superhero.
Superhero, exactly.
Yes,
I'll
because he
be
clearly had a
Hurricane
role
Hazen.
in the X-Men movies. When they cast Richard Dreyfuss, I'll be his doppelganger.
No, very good.
We'll give Dr. Allegretti a Mystique role. I'll create a new character for me, Hurricane Hazen. All right, Howard, you're on.
Okay, we're going to get back to with the rest of the symposium. And I'd like to thank you all for attending and participating. And the next speaker is someone I really admire because he's tackling a question that I think is largely, I won't say ignored, but you don't consider it. For example, if you're eating a yogurt in Paris with the Lactobacillus acidophilus, how do you know if it's the same acidophilus that if you're eating a yogurt in San Francisco? You don't. And you don't know if there's any benefit. You don't even know if the organisms are alive. So Scott Spin, who works about 2 to 3 miles from where I'm sitting right now, has been a real leader in the developing of standards for the microbiome research and product development. This is critical, and I think we were all gratified in seeing the National Institute of Standards and Technology take leadership in this area. So Scott, we look forward to your talk, and thank you for being a part of this.
All right, thanks Howard for that very nice introduction, and thanks to Sabine and others for organizing this. I've been looking forward to it. And it certainly hasn't disappointed. Really great talks from the first speakers. So as Howard mentioned, I am at NIST, and I often feel obligated to introduce NIST because I didn't know what NIST was until I started working at NIST. NIST is a non-regulatory federal government agency in the Department of Commerce. We've got our headquarters just outside of Washington, D.C. in Gaithersburg, where I am right now. But we also have another campus in Boulder, Colorado. We're about 5,000 strong, scientists and engineers. We're the oldest federal technical institute in the United States, over 100 years old, about $1 billion annual budget. And usually when I say technical institute in the Department of Commerce, I get some weird looks, like why does the Department of Commerce need a technical institute? But our mission is to promote measurement science and industrial competitiveness by advancing measurement science standards and technology. So, we really see industry and promoting technology and translating technology as what we strive for. Okay. So, related to that, the industry connection. So, in the last 10 years, we've witnessed an explosion of new biotech and biopharma companies that are all targeting the human microbiome in some way. You heard from a couple of companies this morning, Series and others that are developing the next new modality of drugs, which are live bacteria, but also companies that are exploring diagnostic type services and things like this. So it's a hugely growing industry, and there's a lot of promise behind it, and there's a lot of confidence in that. These things will eventually come to fruition. So last night when I was putting this together, I did a quick search on clinicaltrials.gov for gut microbiome, and there were about 1,300 studies underway that involve gut microbiome, and also did fecal transplant and found about 535. So this is happening quite frequently within the United States and around the world. It's quite a bit of work going on to try to understand how the gut microbiome and human microbiome can modulate our disease. So as I mentioned, the first speakers, Neil and Dr. Wang, gave a really nice setup to this, that poop is efficacious. I like to think that there's magic in poop. It can be highly efficacious at treating diseases. As Neil mentioned, however, some poop works and some doesn't. And people are coining this term super poopers. We don't yet know what the right poop looks like. What is the thing or things that we can measure to determine if an FMT is going to be efficacious? Because right now it's 0. There's nothing that we know to measure a priori that is going to determine or tell us if a stool sample, if a donation is going to be efficacious or not. So the gut microbiome has been described as the most complex ecosystem on Earth. Consists of live microbes and biomolecules, and we need multi-omic approaches to measure this complex system. This is indeed a systems biology problem. And a lot of the woes in the microbiome field are rooted in measurement problems. So how do we measure fecal material? There's many ways really, but the two most prevalent ways are NGS-based metagenomics, where we're looking at the DNA profile of all the bacteria, and from that we're extracting a taxonomic profile to tell what bacteria are there. And the second most prevalent one probably is metabolomics, and this is done either using mass spectrometry or NMR, and it identifies the small molecules, metabolites, lipids, et cetera, that are present. Sometimes these can be quantitative, but that's difficult. But more importantly, these mass spec metabolomic measurements are answering the question, what are the bacteria doing? So metagenomics answers who's there, metabolomics answers what are they doing. In both cases, there are a multitude of methodological variables that impact the results. These are hard measurements to perform. It's been demonstrated many times over that interlab reproducibility is poor. I can guarantee you that if I have a poop sample and I split it in half and I send half to my colleague in Boston and then I measure the other half in my lab, we will get very different results. And that's because we're using lab-specific, lab-adopted methods. The way I extract DNA from my sample might be different than how my collaborator does it. And because of that, we're going to see different relative abundances and different taxonomic profiles. Okay. So there are no agreed upon standardized universal methods for measuring fecal material. As I just mentioned, it's lab specific. So what we need are reference materials to assess accuracy, precision, and to perform inter-lab comparisons and eventual harmonization. If we have a reference material that we know is homogeneous and stable, and that we know 2 labs can have the exact same thing, or 100 labs can have the exact same material, we can begin to understand the extent of the methodological variability's impact on the results. And 2 labs might get the same answer, and they say, aha, now we can compare our data that we're generating in our labs because we know our measurements are producing the same. And contrary to that, if 2 labs measure the same thing and they get strikingly different answers, they know not to really try to integrate or interpret their data in a single swoop. So speaking of reference materials, NIST, we're a metrology institute, the National Institute of Standards. We develop a lot of reference materials. It's not all we do, but it is a big part of what we do. Been doing it since our foundation in 1901. This is a snapshot of some of those materials that have been developed over the last 50, 60, 70 years. You can see that we have standard reference cigarettes, we have peanut butter, we have meat homogenate. Let's see, we've got sulfur and diesel fuel. We've got some industrial sludge, we got Portland concrete. And while all of these may sound silly in some way, like why would you need that? They all have very important industrial applications, and we don't commit lightly to developing a reference material. So if we do develop one, we are going to do our due diligence to know that it has a very specific need within an industry. So let's consider developing a fecal reference material, what that would look like. So people like myself say the words homogeneous and stable at least 15 times a day. A reference material, according to the international ISO definition, is a material that's homogeneous and stable. It's fit for purpose. So manufactured fecal material that's homogeneous and stable. And when I say stable, I mean stable for years, not for months. So we're going to do this once, and we want it to last 5 or 6 years. We don't have the capacity to be, remanufacturing new lots every 6 months. So we're going to produce a lot of it, and it's going to, be that one lot many years. So contradictory to what I just said, poop inherently is inhomogeneous, it's heterogeneous, it's unstable, it's the worst case scenario for a reference material. And just so we're all on the same page, homogeneous means that the microbial taxa and the small molecule metabolites are all the same across multiple aliquots. That's what it would have to look like. Stable means that these metabolites in the microbial taxa don't change over time. So if I measure this material today and I get a taxonomic profile, and then I measure it 2 years from now, that taxonomic profile should not have changed. The ideal preservation method will leave the cells intact and the integrity of the cell wall will not change over time. You can imagine, you know, if the material is being stored for years, maybe the cell wall is gradually being compromised. And over that time, you might think that your DNA extraction method is getting better. Hey, I'm getting better yields, I'm getting more richness, but it's actually the degradation of the material. So these are the types of things that we have to take into consideration and control for. So how do we manufacture large quantities that are stable for years? So last year, we did a feasibility study. We call it the poop pilot study. So we took some poop, we took some water, and we put it in a Ninja blender and homogenized it. Okay, so now we've got a blender full of homogenized poop, and we took some of that and we mixed it with one of these proprietary commercial stabilization buffers. These are, you know, commonly available. These are the reagents that typically you put clinical samples in to keep them stable. So we mixed some of our homogenized poop with this proprietary stabilization buffer, and then we made 4 sub-aliquots of that, okay? There's actually multiple proprietary storage buffers on the market, so we just chose another brand, did the same thing, mixed it with the storage buffer, and we made 4 1 mil aliquots. Okay, then we took some more of the material and we just put it, you know, right as it is, neat, at 4 degrees. No storage buffer. Took some of it, lyophilized it, made 4 replicates of that. And finally, you know, in water, stored at -80. So these are the different stabilization measures that we are testing to see which, if any, will actually lead to a stabilized material over a time frame. And also these preparations have to be fit for purpose. Preparing the material, however we do it, it can't interfere with the downstream measurements in a way that would make it not have the same measurement property. So if it doesn't look like poop anymore, I mean, we could throw some bleach in there and, you know, we could really, you know, hit it hard with some heavy reagents, but, you know, then it wouldn't look like the biomimetic material anymore. Okay, so what did we do with those? Let's see, there's 4 times 5 is 20 aliquots that cover 5 different preservation methods. So we let it sit in the fridge for 4 months or at -80, because one of them was stored at -80. And then after we stored for 4 months, we did 4 types of measurements. We did metabolomics, we did metagenomic measurements, so we did DNA extractions and we taxonomic profiled the material. We did NMR-based metabolomics. We did gas chromatography-based metabolomics, and we did liquid chromatography-based metabolomics, okay? So these are assessing whether or not this material is amenable to these types of measurements. And I'm just gonna give you a high-level overview of what we found. So this is the metagenomic data. So this is the taxonomic profile we found, and it's plotted in a 2D PicoA plot of Bray-Curtis distance. And what you can see is that the different preservation methods all seem to cluster together, which means each preservation method gave its own unique taxonomic profile. And this one that was the material stored at 4 degrees was most different from all of them. And we surmised that at 4 degrees, the bacteria are still alive and there's food and poop, and I would presume they're just, you know, eating the, you know, the nutrients, and they're probably metabolizing and growing, and so that's probably why that shifted away from the rest. But even, you know, within the rest, you can see that there's distinct subclusters that represent the different preservation methods. Okay, so let's look at some of the metabolomics data. This is the GC metabolomics data. This is the, you call it a metabolomic profile. Here's some of the compounds that were identified. And if you look closely, what I'm showing here is that there are 4 traces overlapping each other. And these are for one of the stabilization methods, the 4 biological replicates. So what it means is within a preservation method, whichever one we choose, we see great homogeneity. They're indistinguishable within a preservation method. But you look across preservation methods, different preservation methods get starkly different metabolomic profiles. So this is sort of a storage-specific profile. And those were reproducible. That is, that's different profile from the different storage method. It was reproducible within that method. Okay, go on. This is just a 2D PQA of the GC mass spec data, the one I showed you previously. That was GC, yeah. Same sort of thing. There are some unexpected findings here. So, from the metagenomic data, the taxonomic profile, we saw the 4-degree material appeared to have deviated most from the rest. And in this case, the 4-degree material clusters indistinguishably with the -80 material. So, we're measuring apples and oranges here, metabolites versus taxa. So very different measurements, but that was an interesting finding, and we have some thoughts on why that might be. Okay, and I mentioned that we did LC mass spec and also NMR. It is the same story. We saw great homogeneity within a preservation method, but we saw different metabolic profiles across preservation methods. But more importantly, with what I want to point out here is that these 2 giant peaks that you see in the LC mass spectra and these several giant peaks you see over here, these are the signals from those proprietary storage buffers, and they dramatically interfered with the signal that we're trying to extract from it. So I'm not an NMR expert or an LC mass spec expert, but we do have some of the world's best there, and they're explaining to me that this isn't feasible to use these proprietary storage buffers. They may work well for DNA-based nucleic acid storage methods and downstream, but they don't work for metabolomic measurements because they have such a high concentration of the surfactants and salts and pleiotropic agents that allow it to be a stabilizer. Okay, so conclusions from that. Nothing that you didn't hear me just say. So each preservation method produced a unique metabolic and taxonomic profile. Within a preservation method, the 4 biological replicates were sufficiently homogeneous. So that means our manufacturing process produced a sufficiently homogeneous material. And remember, stool by itself is inherently unstable. So these proprietary storage buffers are not fit for purpose. They interfere with the mass spec measurements for reasons that I had already mentioned. And by the way, we also know that some of those storage buffers, they lyse the bacterial cells. And we don't want that. We want the cells to be intact, because when you do metagenomic measurements, the first step is to do a DNA extraction, and you have to challenge that extraction step. You need those cells intact. So out of those 5, what did we find? We found that the lyophilized material and that material that was stored at -80 seemed quite promising moving forward. And the last thing I'm going to mention, I kept mentioning stability, but I'm sure some of you probably caught on to this. What I just described wasn't a true stability study because we didn't take a time equals 0 measurement. We prepared the material, we put it in a refrigerator for 4 months, and then we measured it once. So there was no T equals 0. But despite that, you could see that we were able to gain some insight from this pilot study. So let's flash forward to 2020. Let's do a true stability study, and we're ramping up our confidence and our efforts here. So in this case, we've recruited two cohorts, omnivores and vegans. It was rather irrelevant what the cohorts were. We chose these because we rationalized that the fecal material from these donors would be most different because your diet impacts your gut microbiome quite severely. So we just wanted starkly different samples. So from these cohorts, we collected their fecal material, we pooled it, we homogenized it, and we made over 1,000 aliquots of it. I mentioned before that we found that lyophilization and storage at -80 was the most promising. So half of these were lyophilized and the other half were at -80. Here are some of our NIST scientists working with scientists from the Bio Collective, which is a stool repository that we work very closely with in helping develop these manufacturing processes. You know, you can see here is a Ninja blender, and if you look closely, you can see some raw fecal material there, actual stool, you know, the homogenization process, there's filtration steps, etc. So my cartoon representation of just a blender is quite inaccurate. Come on. So let's flash forward to March 2020. As I just showed you, we were able earlier this year to successfully manufacture over 1,000 aliquots of fecal material that represent vegan and omnivore cohorts. Now we're ready to start our 18-month stability study. We're going to take our t=0 time point using multi-omic measurements, and we had planned to do, you know, a year and a half time course study on this to really drill down and look at, you know, at individual molecules, metabolites, and ask, you know, there's a lot of volatile compounds in food, you know, and do these things volatilize over time? So these are the types of questions we were asking. However, something happened. It was called a pandemic, and after we manufactured the material, but before we could take these time points, we were sent home. So essentially, we missed our t=0 time point. Sabine had asked me to talk about some of our COVID-related work. I'm going to shift gears here a little bit, but really it's the same thread. When we were sent home, like every other scientist in the world, we were asked to think about how we can respond to this pandemic. And one of the articles I saw early on, I think Sabine actually sent it to me, it showed that this virus was being shed into fecal material. And, you know, light went off in my head. I said, well, we have fecal material, and they're going to be testing for COVID in fecal material, and they're going to need reference materials to, you know, assess, to validate and do interlab studies, etc. So the fact that SARS-CoV-2 is shed in fecal material created some unique opportunities and challenges that we are trying to exploit right now. So one of those is fecal microbiome transplantation safety. Obviously, if a donor unknowingly has COVID-19, they could provide a FMT material that's contaminated with COVID, and of course that could lead to fecal-oral route of transmission. And this was pretty obvious early on in the pandemic in March. The FDA actually released a safety alert addressing this, and they basically said any FMT material that had been donated as of January has to be screened for SARS-CoV-2. Makes sense. So now you're going to have to develop molecular assays that need to be validated in this challenging material. So, you know, a lot of these hundreds of commercial, you know, COVID diagnostics have been developed, but they've all been developed and validated for nasal swabs. So how do they perform in fecal material? And we really don't know that. So we have to figure that out. So we need fecal reference materials, and we have to be in the right place at the right time and have a lot of that on hand. And the second way we started thinking about things, and I'm sure you've heard this on the news, it's quite interesting, and it's really an international push, is to start environmental biosurveillance, so monitoring sewage and wastewater treatment plants. So of course, the fecal material that you flush down your toilet ends up in a wastewater treatment plant somewhere. You can measure community-wide levels of COVID-19 and the rise and fall. And some of the experts say that, you know, this is an early warning system. It's a forecasting system, and this type of visibility would allow public health experts to predict the second wave of outbreaks. Again, sewage and wastewater is not fecal material, but it's not too far from it. So we rationalized that our fecal material could be used in this way as well to help that group begin to validate and standardize measurements for measuring SARS-CoV-2 in wastewater. early on when the pandemic hit, one of my colleagues at NIST, Peter Vallone, he was tasked with getting back in the lab and developing a SARS-CoV-2 reference material. And they did a fantastic job and cranked it out in a couple of months. This is freely available now. You can get it by going on the NIST website. But basically, it's naked RNA in a tube. It's just 2 fragments of RNA that represent 2 pieces of the SARS-CoV-2 genome. These happen to be the regions of the genome where most of the qPCR assays are developed, of course. So it's designed to assess the analytical performance of qPCR assays that have been developed to measure SARS-CoV-2. We also have a lot of human fecal material on hand that we feel confident is homogeneous. And we thought, hey, sounds kind of risky, dangerous, but let's try a spike experiment where we take some of this NIST synthetic RNA and we spike it into the fecal material. And we did that for about 20 spike samples compared to 20 non-spike samples, and it worked. We were very surprised to see that. I was pretty sure that the RNA was going to degrade as soon as it got in the presence of the fecal material, because the fecal material is going to have active nucleases, et cetera. And I thought it was just going to chew the RNA right up, but it actually survived for a week and was still able to be measured. So that seems promising. We're imagining a NIST material that is poop and RNA, and you get it in your lab and you spike one and the other, and now you have your control material. And NIST previously, this was decades ago, but helped develop a standard for synthetic wastewater. Here are the ingredients: includes flour, ocean salts, and beer. They actually use Bud Light, just FYI. But we can make synthetic wastewater in the lab, and you can imagine that for this community, we can begin to do spikes again, or rather, we can hand the community these materials and allow them to do spikes. Okay. I'm finishing up here. I don't know if I'm over on my time.
Yeah, you're a little bit over, but please finish.
Okay. So we had a webinar in June to discuss this with the stakeholder community. It was a webinar on measuring SARS-CoV-2 in wastewater and fecal material. There were about 475 people attended from all over the world. As I said, this is an international effort, and everybody's asking for standards because everybody measures it differently at their wastewater treatment facility, and therefore none of the data is intercomparable. So we're developing a workshop report. It'll be out in a month or two. If you're interested in reading about it, we had subject matter experts come in and talk about it, and really learned a lot. And I'm going to finish. NIST always has postdoc opportunities available. Our programs run through the NRC. It's a very competitive process, but it's a very prestigious process. Our NRC postdocs go on to do great things, including winning Nobel Prizes and becoming the director of NIST. So if you have any talented PhD students, please send them our way.
Scott, why don't you go back one slide and show about what's available, you're making available, the interlay. Yeah, I think you should mention this. This is important.
Okay. So initially, when the initial reason for making the fecal reference material was as a gut microbiome reference material for multi-omic measurements. And just because we're deviating on this COVID road doesn't mean we're giving up on our initial goals and our stakeholders' needs. So with this thousands of aliquots of fecal material we currently have on hand, we are hosting a fecal metabolomics interlab study. We're currently recruiting. If you the URL is at the bottom. If you're interested in participating, you can sign up for it. It's free of charge. We'll send you about 12 or 14 poop samples, and we ask that you perform metabolomic measurements and send that data back to us. And it's going to be an interlab harmonization, and everyone's measurements are going to produce slightly different results and identify different compounds. And it's going to be very interesting to see what we find. So, yeah.
Okay.
Yeah, if you haven't copied the URL, just Google NIST microbiome metabolomics and you'll find it. Okay, we've had a very talented team, a diverse team, everything from analytical chemists to biomedical engineers and everything in between. And that's really what has made this project fun and successful. And these people highlighted in red are from the Bio Collective. It's a stool repository that we've worked with, and they've been 100% helpful in working out manufacturing processes for these fecal materials. And I'll stop with that.
Okay, thank you very much, Scott. That was terrific and a very important area that needs to be continually evaluated. I will take a mention right now. I don't know if for those of you who get Netflix, there's a series called Connections, and episode 2 is on poop. And I would urge you to watch it if you get Netflix. It's worth doing it. But Scott, so this is a little bit outside of what you presented, but what do you feel about standardization of specific microorganisms? Do you think there's a place for that, and how should that be done? Or in other words, I'm talking about how much genetic diversity is, can be accepted. What are your overall thoughts, given you have the broad perspective of standardization?
I think there are reference strains, there are type strains. Microbiologists over the last 100 years have developed like a typing strategy, and there's a pretty strict definition of what a type strain means. I think that's the best we can do, because 20 years ago people thought if you had a strain of E. coli, you just had a strain of E. coli,
Right.
and all E. coli was E. coli. And
Right.
now we know that every strain of E. coli is different than the next, and it's sort of this infinite level of diversity. So I think the best we can do is identify strains that represent the various clades that are that these isolates naturally evolve into, and pick representatives from those. I think that's about the best we can do, and I know some efforts have already gone to do that. Rob Knight and others. Yeah, and this whole taxonomy thing, as Neil mentioned, all we're doing is saying the name of a strain, and how is that supposed to be something that's actionable?
Yeah.
It's like saying my name is Scott, and that's supposed to mean something just because of the name. Like, we need better ways of describing what the microbes are that we're measuring, rather than just stating their names. Which is
I think so, because the genus Lactobacillus contains and I think many of them have been recently renamed because the genus was so diverse that having them all named Lactobacillus was considered inappropriate.
Yes.
So when you characterize the samples, do you also include the fungome and the virome?
Well, we do shotgun metagenomics mostly, so that includes everything except for the RNA viruses. Sometimes we do 16S, so we don't capture that because we don't we don't specifically do the ITS that's done for the fungi. So yes, we see it through the metagenomics that we do.
And what do you consider your biggest challenges in the future?
Oh, getting everyone to perform measurements the same way, that is to adopt a standardized method that everyone is using. That would allow international harmonization of all this data. And just like Dr. Wang pointed out with the cancer studies, those 3 studies, they all found different strains to be what appeared to be the efficacious strain. I guarantee you that's a measurement problem. That's platform-specific, protocol-specific bias that they're seeing. If everyone was on the same page doing the exact same measurements using the same protocols, then we would have international harmonization. But on the other side of that, and this is the caveat, is that standardization stifles innovation. If everyone is doing the same thing, they're not being innovative. So we have to balance that innovation and standardization. And there's really a time and place for both. In a clinical diagnostic setting, there's no room for innovation. You do it the way you're supposed to do it every time. But in a research environment, that's where innovation is born. So, but that's also where the same research is being done. So,
And how much does your standardization, you think, will be biased by the location, where the person actually lives that provides samples, and the sex of the person? So how much variable do you think there'll be in all of that that will make standardization broadly even more difficult?
I don't know how, I don't see demographics being a source of bias in that. What I see is, once the stool hits the collection tube, that's when it starts. And, if we can standardize collection methods, at home, sometimes these at-home testing kits, they just want a cotton swab of stool, and others ask for whole stool. And, I can guarantee you, if you measure both of those, you're not going to get the same result. So yeah, another, phrase that I say 6 times a day is that there's bias in every step of the measurement process, from how you collect your sample to how you interpret your data and everything in between. And there are a lot of things in between. The metagenomic analysis tools that we use, they're called taxonomic profiling tools, and there are these bioinformatic tools that are developed by academics, Kraken, Metaflan, these usual suspects. A colleague of mine and I tracked down all of the various taxonomic profiling tools that have been developed by academics over the last 10 years or so, and we found over 100. There are over 100 different software tools designed to do the same thing, that is taxonomic profile your metagenomic data. They all use different algorithms, they use different statistical assumptions, many of them use different reference databases, and they will all give you a different answer from the same raw FASTQ file. You take one raw FASTQ file and you give it to these 100 different people using 100 different software, and they're going to get very different answers. And that's just one step of the measurement process.
Oh. Okay, well, thank you very much. I think we'll move on to the next talk. And I want to remind the audience that there will be a panel discussion at